Hi, this is GCSE 9 to 1 Crammers. It's never too late to start revising, and this way you can do it anywhere. Listen to these questions as many times as you need until the answers pop into your head. Today we're revising biology, topic 3.2, defenses from infections, monoclonal antibodies and drug trials, covering plant defenses, the body defenses, monoclonal antibodies, and drug trials. Plant defenses. Name 3 physical defenses plants have against pathogens. Leaves and stems have a waxy cuticle, which stops pathogens entering. Cell walls are made from cellulose, which stops pathogens if they get past the waxy cuticle. The outer bark of a tree is a layer of dead cells that stops pathogens entering. Name 2 chemical defenses plants have against pathogens. Some plants produce antibacterial chemicals, e.g. mint and witch hazel. These chemicals kill bacteria. Some plants produce poisons, for example tobacco plants, foxglove and deadly nightshade. These poisons stop herbivores from eating them. Name 3 mechanical defenses plants have against pathogens. Plants have thorns and hairs, which stop animals touching and eating them. Leaves that droop or curl when touched, that knocks off insects or moves away from pathogens. Mimics other organisms, for example passion flowers have leaf spots that look like butterfly eggs. This stops other butterflies laying eggs on them. The body's defenses. Describe 5 ways the body prevents the entry of microorganisms. Skin forms a barrier that produces antimicrobial secretions. Nose hair traps particles, which may have pathogens. And bronchi have lots of cilia, which wafts mucus from the lungs to the throat to be swallowed. Bronchi and trachea produce mucus, which traps pathogens, and the cilia waft it up to the throat to be swallowed. Stomach acid kills pathogens in food. How are the trachea and the bronchi adapted to defend the entry of pathogens? They secrete mucus to trap pathogens. The cilia then waft the mucus up to the back of the throat, where it is swallowed into the stomach. What part of the body is especially designed to defend against pathogens that get past the body's defenses into the blood, cells and tissues? The immune system steps in to destroy them. What's the most important part of your immune system that fights pathogens? White blood cells. Give 3 ways in which white blood cells help defend the body against disease. They consume pathogens using phagocytosis, which are broken down by enzymes into amino acids. Some white blood cells produce antibodies, which destroy bacteria, and some produce antitoxins, which counteract poisons released by bacteria. What is phagocytosis? When white blood cells engulf pathogens and then digest them. After being ingested, the microbes are digested in the cells. Briefly explain what happens to the proteins that the microbes contain. They are broken down by enzymes into amino acids. What protein does the white blood cells produce in the body when they come across a foreign antigen? Antibodies. What are white blood cells called that produce antibodies, also known as B-lymphocytes? Every invading pathogen has unique molecules on its surface. What are they called? They're called antigens. How to remember antibodies and antigens. The body produces antibodies, both end in body, while pathogens produce antigens, and they both end in gens. When do white blood cells produce antitoxins? When the body has to fight toxins produced by invading bacteria. Monoclonal antibodies. Monoclonal antibodies are antibodies that are made in bulk to attach and attack specific protein antigens. What are monoclonal antibodies? They're antibodies made from a single clone of cells. Each type is specific to one binding site on a specific protein antigen. They target very specific cells in the body, for example cancer cells, or a specific chemical, for example the pregnancy hormone. Monoclonal antibodies are designed to lock onto specific antigens on a cell and not onto other antigens on a cell. How are monoclonal antibodies made? Monoclonal antibodies are made by stimulating mouse lymphocytes to make a specific antibody. Large amounts of these specific monoclonal antibodies can be collected and purified. Describe the steps in the procedure to produce monoclonal antibodies. A mouse is injected with a specific protein antigen to form white blood cells that produce antibodies called B-lymphocytes, which are taken from the mouse. At the same time, tumour cells are grown in the lab. The B-lymphocytes are fused with the tumour cells. This makes a cell called a hybridoma. These divide quickly to make lots of clones that produce monoclonal antibodies, which are collected and purified. What are B-lymphocytes? They're white blood cells that produce antibodies. What is a hybridoma cell? When a B lymphocyte is fused with a tumour cell. What happens in a monoclonal antibody production after a hybridoma cell has been made? The hybridoma cells are cloned to get lots of identical cells that all produce the same antibodies, monoclonal antibodies, which are collected and purified. Describe how injecting a specific monoclonal antibody helps a patient suffering with the disease. A drug specific to that disease can be attached to the specific monoclonal antibodies for that disease. The monoclonal antibodies will be injected to the patient, and they will target and find the specific antigens on the cells for that disease. The monoclonal antibodies then bind to those antigens and delivers the drug to kill those antigens without harming any surrounding cells. Name 4 uses of monoclonal antibodies. They're used in pregnancy tests, detecting pathogens to diagnose, plant diseases, human diseases, and animal diseases. They are also used in treating cancer and measuring chemicals in the blood, for example in drug tests. Explain how monoclonal antibodies can be used to diagnose diseases. Monoclonal antibodies bind to specific antigens found on pathogens, on blood clots, cancer cells and certain chemicals. They can also carry markers and fluorescent dyes that make it easier for doctors to see where a problem is occurring. Name 3 side effects that can be caused by using monoclonal antibodies: fever, vomiting, and low blood pressure. What are monoclonal antibodies good at? They are good at targeting very specific cells or chemicals in the body. Name a disease that can be treated using monoclonal antibodies. Cancer. Explain how monoclonal antibodies can be used to treat cancer. An anti-cancer drug is attached to the monoclonal antibodies. The monoclonal antibodies only target the cancer cells and deliver the substance without killing any normal body cell near the tumour. How could monoclonal antibodies with fluorescent dye attached to them be used to identify the athletes using banned drugs? Monoclonal antibodies could be made that will target a specific chemical in a banned drug. Blood samples from the athletes could be tested using the monoclonal antibodies. If the chemical is present, the monoclonal antibodies will bind to it and can be detected using fluorescent dye. In a positive pregnancy test, What does the monoclonal antibody bind to? To the hormone hCG in the urine. Explain how monoclonal antibodies are used in a pregnancy test to see if a woman is pregnant. In a pregnancy test, the stick you wee on has antibodies to the hCG hormone with blue beads attached. If the woman is pregnant, the hCG hormone binds to the antibodies on the blue beads. The urine carries the blue beads up the stick to the test strip. The test strip has more antibodies stuck on it. The beads and hCG hormone bind to the antibodies on the test strip and get stuck, turning it blue. If the woman is not pregnant, there is no hCG hormone to bind to the antibodies on the blue beads. The urine still carries the blue beads up the stick to the test strip, but with no hCG hormones, the blue beads do not bind to the antibodies, so the test strip does not turn blue. Drug trials. Why do drugs have to be tested in a drug trial? To test for toxicity, dosage, side effects, and efficacy. This means how effective the drug is at treating the illness. Explain what testing must be done before the new drug can be used to treat people. Preclinical trials. Laboratory testing of the new drug is done on cells, tissues, and live animals to test the toxicity, dosage, and how effective the new drug is. Then they have the clinical trials. Phase 1. Testing on a small number of healthy volunteers rather than patients, giving very low doses of the drug to see if there are any side effects. And Phase 2. Testing on patients suffering from the illness, to find the optimum dosage and how effective the new drug is. If this stage is successful, they move on to the double-blind trial. Half the group is given a placebo, which does not contain the new drug. The other half of the group is given the new drug. Two groups are randomly chosen, so neither patients nor doctors know who has the placebo or who has the new drug. If the drug passes all the tests in the trials, then it will go forward to a peer review, where other scientists check that the work is valid, and that the drug has been tested thoroughly before being used to make sure that they're safe. What is meant by the term optimum dose? The point where the dose of the drug is most effective and has few side effects. What is the main purpose of preclinical testing? To test for toxicity. What are drugs tested on in preclinical testing? Human cells, human tissues, and animals. Why are healthy volunteers used in the first part of the clinical trial of testing a new drug rather than patients? It might conflict with the patient's treatments. What is given as a placebo? A pill or injection without any of the drug. What is the placebo effect? When the patient expects the treatment to work and feels better, even though the treatment isn't doing anything. What is meant by a double-blind trial? When neither the patient nor the doctor knows who in the clinical trial is given the placebo or who is given the drug. Many people in the UK take sleeping pills. The drug thalidomide was developed as a sleeping pill in the 1950s. It was also effective in relieving morning sickness in pregnant women. This drug was not tested, nor was a drug trial done to see the effects in pregnant women, and it wasn't known that it could pass through the placenta, affecting the fetus and causing abnormal limb defects. In the 1960s, thalidomide was banned because of this. Recently, thalidomide has been used to treat other diseases. Name 2 diseases thalidomide is now used to treat: leprosy and some cancers. In this video, you have learnt about the plant defences and body's defences against bacterial, viral, fungal and protist diseases, how monoclonal antibodies are developed, and how they're used in the body to detect diseases, testing new drugs and the different stages of drug trials before they can be given safely to the public. Take time to learn this topic so you can nail those questions in the Biology GCSE Paper 1 exam. The key to exam success is learning, consolidation and keeping this topic fresh in your mind. How do you do that? Repetition, repetition and repetition. If you go over these enough times, you'll be able to answer any questions on this topic confidently in your exam. This is GCSE 9-1 Crammers, and if you like the content of this video, give us a thumbs up. And if you have a question or need help, give us a comment below. And don't forget, smash that subscribe button if you want more Crammers videos for your exam success. Thanks for watching and see you again soon.